So the first success of gene therapy took place in 1990, and these two girls had a blood disorder, which was also caused by the gene mutation. And so the scientists, they took the bad gene and they isolated it inside bone marrow cell. And so after a while, they injected it back into the girls, and they were healthy. They showed no disorder, no mutation, and they lived a normal, healthy, rest of their life. And that was actually the first success of gene therapy. So what exactly gene therapy is? It inserts a gene into the body so it can help sickness and cure disease. And scientists have stated that in the near future, they can use genes to cure all types of disease without having to go through surgery. So you really don't have to go open up your body, do all the boring, not really boring, the more painful way. You could just do the gene way. So gene therapy, it inserts the gene into the body, but you can't just go directly in there. That's the process I'm going to be talking about right now. So how genes are transferred for therapy to start. So pretty much they put like the good gene that they're trying to replace. It has to go into a cell, which is the vector. But vectors are like used as viruses in gene therapy. So they can actually insert genetic material, so pretty much like the good stuff, the good genes, into certain cells so they can recognize it. So instead of recognizing like bad, like a disease and stuff like that, it can recognize the good side of it, which would be not having the disease anymore. So pretty much that's how they're inserted into there, and that's how you can do gene therapy. So the three approaches scientists are actually thinking of doing in the near future, they're thinking about replacing the mutated gene with a healthier gene. So pretty much like what I was talking about, they take out the mutated gene and they replace it with a healthier gene, so it doesn't have to, like, they don't have to go with the mutation anymore. The gene will be full, it'll be healthy, it'll be functioning correctly, everything like that. Next, they're thinking about inactivating the mutated gene. So pretty much turning off that gene, which would, if you think about it, it can actually stop the disease or whatever's going on with the gene. So you're pretty much just shutting off the gene. And finally, they were thinking about implanting a new gene into the body to stop disease. So pretty much just putting a new gene into your body, and you'll still have that mutated gene in there, but the new gene will go into your body, and it will actually pretty much target that disease and take it out, so you won't have the disease anymore. The pros and cons of gene therapy, that the pros are, it can give someone with cancer a normal life. And pretty much, like, you can have cancer, the gene therapy, you go through the whole thing, and you'll live a normal life. You won't be like, oh, I have cancer and stuff like that. You'll live the normal life. And so, like I was saying, you can also get rid of disease in the body. So pretty much you can rid the disease and live the normal life. But the cons, like completing gene therapy, getting it done, it's very risky, and that can lead to very serious disease that will be caused by like not going correctly 100%. So it's actually still under study to make sure it's 100% safe. So like even if it's 99% safe, that 1% can completely mess up your entire gene therapy, and it can give you more disease. And that's why it's only been tested on disease that have no cure. That's why it's the con. It's only tested on disease that have no cure. So you don't know if it works on like a disease that has a cure. So you don't know if gene therapy might be a quicker, more affordable way. You don't know that yet. So that leads to the final thing, the ethical issues. Like, these are the questions, like, how can good or bad issues be determined? How can they say that doing this process is good, doing this is bad? Because you never know. Mistakes might lead to making better things. And who decides which traits are normal and which aren't? You don't know. You really don't know which ones are good, which are bad. It's just you're assuming. Like, some you can completely tell which are good, which are bad, but some you really can't. So, will the high cost of gene therapy only be available to the wealthy? So, like, gene therapy is predicted to be very expensive. So are they only targeting the wealthy for that one? Because what if you can't afford it? What are they going to do about that? And so could widespread use of gene therapy lower society's acceptance of people? So like they might think, okay, you did gene therapy, like they might not accept you for who you are anymore. They might expect like lower standards. And should people be able to use it to add height, athletic ability, stuff like that? I really don't think that they should be able to, because why would you do that? It's like taking steroids. Why would you do that? So you can be taller, more athletic? Like, I really don't understand why people would do that. So that is one of the questions that is very questionable. So yeah, that was gene therapy. So right here is a diagram of how like cells pretty much go through gene therapy. So first off, cells, they are removed from the person who has the mutated gene, and in the lab the virus is altered so it can't reproduce. So that virus that keeps on spreading and spreading like cancer cells, it's like completely changed so you can't reproduce anything, so it can't grow. And then next, the gene, like the good gene that will kill the virus, it's inserted into the virus, and this altered virus, the one that can't spread, it's mixed with cells from the person who had the bad genes. And so then the cells from the person, they become genetically altered, and the altered cells, pretty much they are, once they're completely good and they don't have the mutation in them anymore, they're injected back into the person. And the cells, they produce the protein or the hormone which will prevent that disease or whatever from growing. So you might have the mutated gene, there might be two of them or something, and you have one of them, it might go and it'll try to find that one gene that is, like, it'll kill it. So it'll prevent any more disease like that from spreading.
Virus particles containing the therapeutic gene enter the liver with the bloodstream. Their target is the region where liver cells exchange matter with the blood, the small capillaries. There are large pores in the capillary walls. The liver cells are on the other side of these openings. The capillaries also contain special cells, the macrophages. Their task is to remove any foreign and harmful particles. They will try to do this also with the virus particles. That is why the outside of our virus is coated with a protein that can bind specifically to liver cells. Because of this protein, the virus will get past the macrophages and reach the liver cells more quickly. Once the virus particle binds to the liver cell, the virus's envelope merges with the liver cell's cell membrane. This allows the virus's load of therapeutic DNA to penetrate the cell. It will find a path to the cell's nucleus. The virus proteins assure that the therapeutic gene is taken into the genetic material of the liver cell. This will enable the liver cell to function once again as a healthy cell.